lots of outputs from these 16 box Punnett squares. And exactly how you get those outputs isn't always explained to us. So, for instance, we've looked at this one where we get 4 different outputs from 2 genes coming in 2 allelic forms. And how we get these outputs depends on whether you're dominant or recessive at each of these 4 genes, or these 2 genes. But we're not given a biochemical explanation. We're just being told that, well, if we have these kind of generic A and B terminology for the gene, and you're a certain genotype, then you get this. And if you're a different genotype, then you get Y. Of course, underlying this is the idea that genes produce proteins, and then they have an effect with one another to control development and so forth. Another example is here. They've designated the genes A and B, and depending on what arrangement you have for the wild type big A and big B compared to the recessive versions, you generate a certain developmental output, and that's going to depend at the biochemical and cell biology level on what these genes do and how they control things and so forth. So for some of these examples, we're given a kind of the genetics of it. We're shown how the genetics works, how it plays out, how we determine phenotypes based on this 2-gene model, but we're not told kind of the answer, the underlying biochemistry of how it's working. In some of these cases, it may be known. In other cases, it may not be known. So there's some ambiguity then for some of these examples as to how they work. So I just want to point out that in all these cases, these genes are producing something. It might be transcription factors that regulate other genes, right? It might be enzymes that control metabolic pathways. And then you have to consider whether you're making 50% of a certain protein when you're a heterozygote. Or maybe you have to consider that the recessive allele has some different activity. Things like this. And then these 2 alleles on these 2 different genes will combine in certain ways to generate an output. And we're not always told exactly how that's working. So I want to go through another example of a 2-gene system where we generate yet another proportion of outputs from that 16-box Punnett square. And in this example, we have some plausible biochemical explanation for what these genes are doing and how they're causing the ratio that we observe. And this model has to do with pea plants again, and it has to do with flower color. So the idea here is that we start with one parental true breeding strain of pea plants which produces white flowers, and we start with another different strain of pea plants which also produces white flowers. But lo and behold, we cross these and we expect white flowers, but we don't get white flowers, we get purple flower. So this is a surprise, right? It's contrary to our expectation. We have no reason to suppose that in this cross we're doing anything other than, let's say, self-crossing this, in which case we get white, or self-crossing this, in which case we get white flowers, right? But here we take two plants which seem to be genetically the same, producing the same output, and we get something totally different. And we've seen this again and again in some of these examples that we looked at. And we know the answer genetically, it's already been worked out. So what they've done is they've assigned us genotypes for the parental plants to begin with to make the problem kind of convenient to go through. And so this one is dominant at one gene, recessive at the other, and vice versa. This is recessive at the first gene and dominant at the other, and they're homozygous, so they're true breeding. We cross them, we get this F1 hybrid, which is big C, small c, big P, small p, and for some reason this is purple. And then again, what we do in all these cases is we take the F1s, which are heterozygous, we cross F1 against F1 to generate our out of 16 set of boxes in a Punnett square, and we apply this branch diagram approach and try to determine what the genotypes and phenotypes would be. So as before, we treat the C and the P genes as independently, and so we know from the first cross 3/4 are big C something, of those 3/4 big P something, 1/4 small p, and likewise for this category, 1/4 of the cross of big C times big C small c will be small c, and I've said this again and again, but of those, 3/4 will be big P something, and 1/4 will be small p. And we multiply everything out in the branch diagram, and we get this same set of 9, 3, 3, 1, but we're going to apply different rules to this one compared to what we've done for the other ones. And in this particular case, this category being dominant for at least one allele of both the C and P genes gives us one output. Being recessive at one, or recessive at the other, or recessive at both generates a second output, which we are going to kind of cluster together to form a different phenotype, and we get a ratio 9:7, something completely different from those out of 16 examples that we've seen so far. So there's a lot of variation that you can get with just two genes coming in two allelic forms, depending on how the genes interact with one another at the biochemical level, and depending on what they do. And I want to point out also that when we do this type of branch diagram, we're making some assumptions, right? And it's good to keep in mind what those assumptions are. One assumption is big C is dominant to small c, and big P is dominant to small p. So what that means is we can say an organism would be big C something, and we don't care what the something is. It could be another big C allele, or it could be small c. It doesn't make any difference. We're going to see the big C output. Okay, so that's one thing that is illustrated by the dash there. And the other thing is that in all of these examples, we're treating the C gene chromosome separately from the P gene chromosome, or A from B, and so forth, and other examples. So we can assess the output of this cross independent of the output of this cross, and then multiply them together. That's a kind of particular Mendelian thing we can do. It's not something we can do all the time. We'll see exceptions to this later. But in these examples, we are always treating this first gene cross independent of the second gene cross. So things to keep in mind when we think about this branch diagram here. And maybe the last point is we're looking for phenotypes in these branch diagrams that were being shown. And so we don't care whether it's big C, or big C, small c, because of the rule of dominance and recessiveness in this case. The output is going to be the same either way. But there's going to be problems that you work, or maybe you do have to know exactly what the genotype is, or maybe there's a difference between big C, and big C, small c. So in those cases, your branch diagram would look a little different. So let's suppose you did a cross big C, small c times big C, small c, and you cared what the genotype was exactly, because maybe the output phenotype would depend on the genotype. So in that case, you might have 1/4 big C, 2 out of 4 heterozygous big C, small c, somewhere in the middle, and another 1/4 small c. So there are branch diagrams that you would have to set up, and where you kind of partition this into each of the types of genotypes that could be produced, rather than make the assumption that big C is always dominant to small c. So for instance, in those Andalusian chickens, if you had one set of alleles, you were a dark colored chicken. If you were heterozygous, you were an intermediate, a kind of blue colored chicken. So that would be a case where you might not want to do that problem in the same way that you see here. You might have to have 3 categories. Okay, so now, how do we explain something like this? Now, this is, I think, a neat thing. Let's suppose that we can explain this coloration pattern by supposing that there's a metabolic pathway where various compounds are interconverted amongst one another through some pathway, and that pathway is controlled by enzymes, which are encoded by these C and P genes. So let's suppose that there's a pathway where we have a chemical which is white in color, or no color, whatever, and that chemical can be converted to another chemical which is also white in color, and that happens, let's say, in the flower. And this chemical too can be converted to a purple colored chemical. So you could have different colored flowers depending on kind of what you had in the flower in terms of these different chemical compounds. And so here we have a metabolic pathway. where this chemical converts to this one converts to this one. And let's suppose that in the genetic system that they showed us in the previous picture, let's suppose the C and P genes encode enzymes which catalyze steps in this metabolic pathway, right? This is an important idea in genetics, the idea that genes control enzymes which then have some biochemical effect. So we'll say that gene C would be located here. Gene C encodes a protein C, let's say, which is an enzyme which converts this chemical to this chemical. And let's suppose that gene P encodes a protein or enzyme P, which is able to convert this chemical to the purple chemical. So now we have assigned genes at a location, specific location along a biochemical pathway. And this is an important concept and lends itself to lots of problem types and so forth. Okay? So if we propose that this is the situation, then it kind of makes sense how we're going to get out those phenotypes from that the Punnett square. So we assume big C is dominant to small c, and usually what we mean by this is that big C is functional. It makes a protein that works, right? It's an enzyme that's active. Small c, the recessive allele, is in many cases defective or not produced at all, something like this. Okay, so we'll assume big C is dominant to small c, and big P is dominant to small p. So given all this, we could see that if an organism was homozygous small c, they would not make any good enzyme C to catalyze this step. The plant would be blocked right here because they would be defective for both alleles of that C gene, so they'd be blocked here. This chemical would accumulate, but it would never move further down the pathway. Okay? In which case the flower would be white colored because this chemical lacks color, it's white. If a plant were small p, you'd get a similar effect. In that case, this step is defective. You don't make any good enzyme P, so you're blocked in this metabolic pathway right here. So even if you can convert white color 1 to white color 2, you're blocked here, and so you accumulate this intermediate chemical, which is white, and you do not make purple flowers. And the only way that you could make purple flowers then would be if you had at least one good copy of gene C and at least one good copy of gene P. That would give you enough protein enzyme to catalyze the conversion of this to this, and then this to this, to end up with purple. So these types of problems are neat because they let you assign metabolic steps to particular genes and assess the output and phenotypes based on this idea of each gene controlling one of these steps in the pathway. So you'll see problems like this in the book. So you need at least one good C and one good P to go to purple, and if we go back to our little table here we have one good C, one good P at least, and so you went all the way to the end of that pathway. In this case, your P gene was defective. In this case, your C gene was defective. In both of those cases, you stop the pathway at some point and don't go to purple. In the last case, you're defective for both, so the outcome is kind of the same. So pay attention to these types of problems. They're interesting problems. One thing that comes up once in a while is people will say, well, I have a genotype and it's small c, but the P gene is good. It's big P dash, let's say, okay? So why don't I go to purple? It's easy to kind of confuse yourself and say, well, you're going to go to purple because this is good. If this gene is bad, then this is not present at all. So even if you have a good copy of the P gene, if this gene is defective, you stop the pathway right here. You never produce the substrate for the P gene. So don't confuse yourself that if you have a good copy of P, you necessarily go to the end. This is kind of like the blood group thing, where you have to have the H antigen present for IA and IB to act on it going in one direction or the other direction. If you don't have H in the first place, the presence of IA and IB makes no difference whatsoever. There's no substrate they can work on. So the same kind of logic applies here. Okay? And this is a simple pathway. You can make more complicated pathways, right? You could make branches and all kinds of stuff. And it starts to become a headache, but nonetheless, this is the basic concept here. So I like this one because you get a plausible biochemical model for how you arise at this 9:7 ratio. Okay, so I think the last topic in this extensions to Mendelian analysis stuff has to do with lethal alleles, and this is pretty interesting. We saw before a picture of a yellow mouse, right? And they're nice looking mice. And the question is, what's the genetics that underlie the production of yellow fur compared to agouti or black or brown or something like that? And it turns out this agouti gene doesn't have just two alleles, big A and small a, but there is yet another allele called AY. And this AY allele at the agouti gene is responsible for making mice that have yellow colored fur. Okay? And another characteristic of these yellow mice is that they don't breed true. And when something doesn't breed true, that's often a hallmark that it's heterozygous, right? If you make two things that breed true, they only contribute the same type of alleles to the gametes, and those only are recombined in the same genotype as the original parents, and so that's what you get over and over again, generation after generation. It's important to remember that when things don't breed true, it's often a signal of heterozygosity for some gene that you're looking at, right? So breeding true means an allele is homozygous. Not breeding true means there's something wrong with that idea, and heterozygosity might be the answer. So that's a characteristic of yellow mice. They don't breed true. Like those Andalusian chickens, they don't breed true. So if they don't breed true, and if we assume that these mice are heterozygous, what happens when we cross a yellow mouse against a yellow mouse? Not breeding true means you don't get just yellow mice out again, but we could say that if we started with an animal that's AY and big A at the agouti gene locus, and another animal that's the same genotype, AY and big A, that would be our heterozygous starting state. What would we get in a cross like this? Well, this is a simple Mendelian cross, like smooth and wrinkled and so forth. You'd get AY plus AY, AY plus A plus AY, A plus A. So here's the set of genotypes that you ought to get in this case if we suppose the starting animals are heterozygous. We get one part this, one part this, one part this, and one part this. And that's fine and good. The problem is when you actually score the offspring from a cross like this, what you observe is 2 parts yellow progeny and 1 part non-yellow progeny. And we have to kind of figure out what in the world is going on there, because we expect an out of 4 set of offspring, right? This is a really simple breeding experiment, and there's 4 possibilities. But we don't get an out of 4 set of progeny, we get an out of 3 set of progeny. Okay? And the explanation is that 3 of these embryos after fertilization are viable and will develop to birth and into adulthood, but 1 of them is not. The AY-AY allele combination is lethal, embryonic lethal. So even though we may get these fertilization events occur early on, those embryos will die due to embryonic lethality, basically miscarriage. Okay, and so what actually continues to birth will be the other 3, and that's why we get this. Excuse me. This out of 3 output of progeny in this cross. Okay, and the fact that these die explains why the yellow mice don't breed true. They have to be AY something else. You can't have true breeding AY mice. Okay. So the picture at the right is from the book, and it just shows you various crosses concerning this agouti gene and the A and AY alleles. Here we have true breeding agouti versus agouti. You'll always get agouti. Here we have the cross that I just set up over here. We get 4 different genotypes produced initially. One of them is lethal, the other 3 make it. So we get, 1 part agouti, 2 parts yellow, and 1 lethal to produce this out of 3 ratio of offspring. And the cross on the right-hand side is just the yellow mouse against an agouti mouse, which is homozygous for that agouti allele, and we get half agouti, half yellow. Okay, so it's kind of again, one of these kind of funny things that happens, gives us a skewed ratio of offspring that we don't expect, something that's non-Mendelian, where we expect an out of 4 set of offspring, it's out of 3, and the explanation has to do with lethality early in embryonic development. So the next question is, why does that happen? And that gets to the kind of molecular biology of this particular system, and so I want to give you an idea of what happens in these AY mice to cause that full yellow coloration of the fur, and why it's lethal when homozygous. Maybe before I do that, I'd like to just point out that if you had a colony of mice and it just so happened that all of a sudden a yellow mouse appeared spontaneously, you might say, well, that's a weird mutation in some gene in the genome, and it's worth to investigate to figure out what in the world is the cause of that yellow fur color, okay? And you have a lot of possibilities to explain why the fur color might be yellow. And one of them would be, maybe there's some other gene you didn't know about, some separate yellow gene, we could call it, that when mutated causes this yellow fur coloration. That's a perfectly good explanation. Why not have an agouti locus, a black-brown locus, a coloration C locus, and a 4th one that controls, some accidental yellow fur coloration? Perfectly good explanation. You don't know. What you have to do is lots and lots and lots of breeding experiments this way and that way and so on and so forth over a lot of time and figure out really what's going on genetically. And it just so turns out that the reason for the yellow fur is due to an allele at the agouti gene itself, okay? So this isn't something that you would just know by looking at a mouse saying, oh, it's got to be another allele at that agouti locus. Not necessarily. You have to do experiments to figure that out. So don't think that you can just kind of look at something and guess necessarily. Okay, so why is AY causing yellow fur, and why is AY lethal? So what I have at the bottom here is a depiction of the normal genomic arrangement of genes near this agouti gene. And in a normal chromosome in a mouse that is agouti, we have a gene agouti which encodes a protein, and this protein is responsible, if it's the big A allele, this is responsible for making that little stripe of fur yellow against a background of black or brown fur. So that's what this gene does, and it's located here, makes a messenger RNA, produces a protein, that protein causes that yellow band of fur. Near the agouti gene is another gene called Merck, okay? The Merck gene is another mouse gene, and it has to be intact and complete, and it will produce a Merck protein, and it does whatever it does. I think it's an RNA binding protein, but it doesn't really matter what it is. But it's an adjacent gene located near the agouti gene locus, okay? So genes that are next to one another along chromosomes typically don't have anything to do with one another. So there's no reason to think that Merck affects agouti or agouti affects Merck. Genes are located kind of randomly throughout the chromosomes. So these just happen to be next to one another, okay? So on a normal chromosome, agouti gene produces this agouti protein, and the regulation of this gene is due to promoter and regulatory sequences right here. The Merck gene produces Merck protein, and how that's accomplished is due to regulatory sequences like promoters and enhancers and so forth for the Merck gene. And everything is fine and good if you have this chromosome arrangement. In the case of the AY allele, what's happened is a big section of DNA between the Merck and agouti genes has been deleted, right? That's a common type of chromosomal problem or rearrangement and so forth. So there's a big chunk here that's gone. And so now the chromosome with the AY allele looks like this. There's been a fusion of the Merck and agouti genes. This fusion still allows the agouti protein that's produced from this kind of hybrid or chimeric gene. The agouti section is still active. It can still produce that yellow coloration, that stripe on the fur. And the problem is the agouti gene, though, has lost its regulatory sequences, and so the regulatory sequences that are now driving agouti are the Merck promoter enhancer sequences. And Merck is on all the time. Agouti should be kind of on and off just to make that little stripe. But now you've linked a functional agouti gene to the regulatory sequences that drive expression all the time. So you're producing lots and lots and lots of this kind of hybrid protein, and it's acting actively to produce yellow fur coloration along the entire length of the fur. It's no longer restricted developmentally to just produce that little stripe. So deletion results in the loss of these agouti regulatory sequences. You produce a hybrid protein that's still active for yellow fur coloration, and so the mice appear yellow. So that's pretty interesting. And then the next question is, why is the AY allele lethal? Okay, and the answer is, in this allele, AY, you have the agouti gene activity linked to the Merck regulatory region, but you've lost the Merck protein. You've deleted enough of Merck that it's no longer active. Okay, so you don't get any functional Merck protein in this arrangement. Okay, and if you have two copies of this AY like this, and then another one just like it, you don't produce Merck at all, and Merck is required. It's essential. So being AY means you have no ability to produce Merck, and you need that for development. You don't have it, and so that's why it's lethal. AYA mouse would have one copy of a chromosome maybe that looks like this, so they would produce agouti correctly. They will produce Merck correctly, right? And they will have another copy of a chromosome that looks like this, which is AY. It doesn't produce agouti correctly. It produces it all the time, so this is dominant to this. This activity overrides the little stripe that should be produced. It doesn't produce any Merck, but you still get Merck from the other chromosome. So the heterozygote AYA is good because you do produce Merck. Okay. Make sense? It's kind of a slick little example of what's going on with two genes. It starts to give you a connection between just genes as letters like A and B and whatever, to genes as proteins and genes as real structures in the genome, I think. Okay, so last thing here is just a few kind of side notes if you're interested in other stuff. What about human hair color? Well, what is there? There's red, yellow, brown, black, etc., right? So that's all interesting. And if you want to learn more about the genetics of something like that in comparison to, mice coat color or Labrador coat color, look at Wikipedia. There's a little interesting article here. It also involves dyes, right? So there's other things. You can buy all these hair color dye products in the grocery store. And it involves things like wigs. So there's other aspects, non-genetic aspects as well. So if you're interested, take a look at the article. Some of us are not interested, right? It just doesn't matter anymore. Okay. And then what about eye color? Eye color is another big kind of obvious visual feature that people are interested in genetics. The rule of thumb about eye color is always that the brown causing allele is dominant to the blue allele, because with brown you produce a pigment, right? You produce something, whereas not brown you don't. So that's kind of a common rule of thumb. But on the other hand, everybody knows that there's all kinds of shades of eye color, right? Ranges of blue, ranges of brown, all this kind of stuff. So there's lots of genetic influences on this, and people study this. Okay, so here's just some interesting examples. Here's a cat that's got two different colored eyes. Okay? I got it off the internet, so I'm assuming it's not been faked or whatever. But, okay. And then here's another example. This guy is a baseball player, right? His name is Max Scherzer. He's a pitcher or something, and he has two different colored eyes. And this is called heterochromia. And I don't really know what exactly underpins this type of a thing, but you could look it up if you're interested. Here's David Bowie, he has 2 different colored eyes. Here it's not genetic, it was an accident, okay? So he had, somebody hit him when he was a kid, I think, that's what I understand. And so somehow one eye is different from the other eye. And there's colored contacts. This is not strictly an immediate genetic effect, but nonetheless, there's this option. And then you can give yourself whatever eye color you want if you take a picture of yourself and edit it in Photoshop. So again, not genetic, but still kind of interesting types of things. If you wanted to know more about this, again, look at Wikipedia or these other sites, and they'll tell you lots about these things. People study them. It's a very interesting type of phenomenon. Okay, so that's it for extensions to Mendelian genetics. Things like multiple alleles, codominance, incomplete dominance, and a bunch of these 2 gene systems where we generate 16-box Punnett squares, but we get this output or that output or another output, depending on how those alleles are combined in an organism to produce some effect. And for most of them, we don't get an idea what the underlying biochemistry is. We don't know whether the proteins metabolize hormones or metabolize pathways or are transcription factors. We don't know, but at least for one of them, the pea flower color model, you do get a sense of connecting a gene to an enzyme to a metabolic step. Okay, so now what? Now, quantitative genetics. So quantitative genetics, it sounds a little bit disturbing, but I will try to fuzz over a lot of the quantitative parts and all these formulas and stuff so that we get a sense of what genetics we're talking about here. And usually quantitative genetics in an intro genetics book is a chapter that's often put off to the end of the book, right? And as students, you know, those chapters at the end of the book, you never get to those, but we're going to get to it, okay? And we're going to get to it now because it's really an extension of Mendelian genetics, and it's super important in understanding how you generate really complicated phenotypes, complicated outputs. And to make complicated outputs, you need lots of genes interacting with one another together. The Mendelian situation is really simple. It's 1 gene, 2 alleles, you get this or that. That's kind of artificial. I mean, our genomes are enormous, and they, all the genes combine to generate complicated outputs. So we're going to cover, at least superficially, the ideas present in this particular chapter. Okay, so far, what did we look at? We looked at a really simple situation where you have 1 gene, 2 alleles. We assume dominance and recessiveness, and you get 2 outputs, like yellow and green. That was one super simple case of how a gene controls a phenotype. Another case would be the chickens, where you've got intermediate color, right? Incomplete dominance, where you've got 3 outputs from a 1 gene system with 2 alleles. But the point is, you get either this. It's kind of a binary switch. You get either this category or you get that category. It's real simple. Okay? If we have 2 genes, we start to get more possible phenotypes produced. And we've seen all these examples of these F2s where we got 9331, 943, 961, 97, all these things. A little bit more complicated situation compared to one gene, 2 alleles. And the other thing we know, because genes come in multiple allelic forms, that creates additional phenotypic complexity. For 1 gene, you might have more than 2 alleles. You have a whole range of possible alleles. But in any given organism or any given person, you only do get 2 of those, right? Because we're diploid. But the pool from which you can select might be fairly large. So you do get more phenotypic diversity if you consider the fact that there's got to be multiple alleles. But when you ask yourself the question, can you take 1 gene or 2 genes, and can you make something really complicated, like an ear or a foot or something? Is there an ear gene? No. Is there a foot gene? No. You need all your genes acting together collectively to produce a particular output in these types of complex phenotypes. Okay, so that's kind of type of thing we're going to be thinking about in this chapter on quantitative genetics. Okay, so far we've seen phenotypes that come in discrete forms, yellow and green, smooth and wrinkled. and they could be in a 3-to-1 ratio, like those Mendelian pea plant experiments, or the Andalusian chickens came in a black, a gray, and a white 1-to-2-to-1 type of thing. But in both these cases, you have discrete phenotypes. You look at an organism and you say, yeah, I can put it in this bin or this bin or something like that. Although I have to say that if you read that paper by Mendel, that translation, he makes a note in there that even for things like yellow and green, they were still kind of more yellow or yellowy, or I put it in yellow, and more green or greenish, things like that, and I put it in green. He had still to make decisions, even in that case. It wasn't as strict as the graph like this would lead you to believe. Okay, so there was still some kind of either genetic or environmental fuzziness that still influenced greenness and yellowness, even in that simple 1 gene, 2 allele system. It could still be, because, like in the Drosophila, I mean, I don't know specifically in the pea plants how the greenness or yellowness is caused by the amount of dye and so forth. But it seems to me plausible, like in the case of the Drosophila eye color, you're still red when you got 0.5, right? But if you vary the 0.5 up or down a little bit, you're going to start to impinge on that pinkish area, right? So it's possible that you get an array of green and yellowness as a consequence of heterozygosity plus other environmental or genetic effects. Okay, so here we're going to look at continuous variation, complicated traits that vary a lot between them. You can't really bin them into these discrete categories. So for these types of things, we can model them according to a normal curve, which I've tried to draw here. And we have a kind of quirky example from the book over here. For whatever reason, what they've done is got some data about the birth weight of babies born to teenagers in Portland in 1992. You can't get more unusual or detailed than that almost, right? That's a pretty odd thing to pick. But the point is, if you collect this data, and you bin these babies in different weight categories, you see some sort of a graph that looks like this, where you have, lightweight babies and heavy babies, and they fall into some phenotypic curve that looks more continuous rather than those discrete curves that we saw with those simpler systems. And so we're interested in how genes collectively result in these types of phenotypic curves rather than those discrete curves. So curves like this are continuous, and the reason is you've got lots and lots and lots of alleles in the genomes that are kind of adding up, or each contributing a little bit, ever so slightly, so that individuals over here may have a collection of alleles that are contributing to a little bit lighter weight. Individuals over here are having a collection of alleles that cause a little bit heavier weight. Now this is complicated because you can have maternal influence, you can have environmental effects and so forth, and those types of things are going to smooth it out even more. Okay? But when you have the contribution of a lot of alleles, you're going to get continuous curves that look like this. Okay, so this is kind of the subject of quantitative genetics. The last point is we'll talk about environment. Environment can smooth things out even more as well. So the first example here is to illustrate the idea that if you consider multiple genes and consider the idea that they add together in some way to generate a phenotype, you start to generate this continuity in phenotypic curves rather than those discrete examples. So one of the first examples of this was this polygene hypothesis having to do with coloration in wheat kernels. And this was a long time ago, obviously, more than 100 years ago. And the idea here is that you have some strains of wheat whose kernels are either red or white. And these strains of wheat are true breeding. So if you cross red versus red, you always get the same red. If you cross white versus white, you always get the same white. Okay, so that means they're homozygous for the alleles that are causing red coloration or white coloration. And when you cross red kernel versus white kernel wheat, you get an F1, which is intermediate. That's kind of like the Andalusian chicken example. So you get some contribution of alleles from this parent which are causing coloration, but you don't get the whole set. You get some contribution from this parent of alleles that don't cause coloration, and this is a kind of half and half arrangement here in the F1, so it's intermediate. And we cross the F1s as we always do to generate F2s, and we ask the question, what's the ratio? And the ratio here is 15 to 1, reddish to white. 1 part white, 15 parts some shade of red or pink. So yet another out of 16 in this particular example here. And if you kind of parse out the 15 into different shades, it turns out they fall into this kind of a semi-continuous curve of 1 to 4 to 6 to 4 to 1. So if one of these is red, the 1 part red, the other end is white, 1 part white, and the ones in the middle are kind of intermediate shaded. Okay, so it's, again, another out of 16 ratio that we get. 1 to 4 to 6 to 4 to 1, from more red to less red all the way to white. And we can come up with the model after lots of work, right? We come up with the model that can explain something like this. And we have some details about this model here. I have an explanation in the left-hand column here, and it uses terminology R and C for the genes responsible for it. On the right, I have a Punnett square illustration of the same thing, but the Punnett square illustration that I could find uses a little bit different designation of the genes. It makes no difference, it's just a question of whether you call something A and B or C and D or something like that. So here we use R and C. In this picture, they use R and R prime to indicate 2 separate genes. Okay, so it's not a big deal, but just that's the reason why the nomenclature isn't matching here. So let's go through this part because it kind of explains what's happening in this wheat kernel coloration model. So the parents are true breeding, and one is fully red, one is completely white. Okay? So since we know the answer, we're going to say that the full red parent is homozygous big R, homozygous big C, and all 4 of these alleles of the R and C gene are contributing a little bit to the redness phenotype. And we're going to say the other parent is also true breeding, so it's homozygous at these 2 loci, small r, small c, small c. There's no capital letter allele here, and we're going to say that these are all producing no red pigment, right? They're defective in some way, so they don't produce anything. So that explains why you get full redness from this parent and full non-redness or white from this parent. Okay? And they're homozygous, they're true breeding, so you can only have one possible product in the F1, which would be big R, big C, small c. So all of the F1s look like this, and they will be intermediate in color because they have 2 capital letter alleles contributing to redness, as opposed to this parent, which has 4, and this parent, which has 0. So you get an intermediate pigmentation compared to the parents. And now we cross the F1s against F1s, and we get a whole range of genotypes in those 16 boxes, right? The genotypes range from the original big R, big C, like here, all the way down to the other grandparent, which is small r, small c. Plus you get a whole bunch of genotypes in between here, which have various numbers of big R and big C alleles. And we determine the color in these quantitative genetics problems by kind of just adding things up. We make the simple assumption that there's lots of background alleles, and we can say everybody's contributing a little bit. So let's suppose, in this case, all the capital letter alleles are contributing more or less equally to coloration. If you have more, you're more red. If you have intermediate, you're middle red, and if you have none, then you're not red. So we can say color is determined by how many big R and big C alleles are present. So big R, big C, that's the max red. If you have 3, and there's a bunch of different ways you can get it. Big R, big C, big R, big C, big R, big C, so on and so forth. Lots of different genotypes have that combination where you have 3. they're less red than max, but still pretty red. Then there's some organisms that have 2 copies of R and C, and some organisms that have 1 copy, and so forth. And by doing that, we generate something that looks like this, where, as I said, the designation is R and R prime, but you get the idea that one of the F2 offspring will have a genotype with 4 of the red alleles here. Another one out of 16 will have none of those red causing alleles, and the other ones will have either 3 or 2 or 1. So here's 1 part red. This is 1, 2, 3, 4 parts out of 16, kind of less red. You've got 1, 2, 3, 4, 5, 6 parts out of 16, which are perfectly intermediate. You have 1, 2, 3, 4 parts out of 16 that are going towards the white, and then you have 1 part which is white completely. And that, what you see here, matches kind of this curve right here. So it's out of 16, 15 to 1 red or some shade of red to white, but we can kind of subdivide that 15 into this curve right here. And so you start to see continuity generated in this phenotype, all the way from maximum redness to maximum whiteness, and it starts to smooth out. And that's kind of one of the goals that we're thinking about in quantitative genetics. Okay, it turns out, as I understand it, the system later was analyzed more extensively, and it was determined that it's actually 3 genes, RC and something else, we might say. Here we've designated the 3 genes responsible for the system as A and B and C, and they've given them either a prime designation, meaning they're inactive for red pigmentation, or not a prime designation for the allele that is active. And if you have 3 genes, your Punnett square gets really big, and you can't do one of these on an exam, okay? You just can't. You'll have to do some sort of branch diagram approach. Here's a Punnett square with, 64 boxes. It takes a long time to write out something like this, right? So it's not a feasible approach to analyze a 3-gene cross using a Punnett square like this. But here we get it visually, so it's kind of nice to see. But let's suppose there's 3 genes that contribute to redness, just as in that previous example where I just showed you 2. So now you have a possibility of a maximum of 6 alleles causing redness, or 6 alleles not causing redness. So now you can have 6 or 5 or 4 or 3 or 2 or 1 in proportions that depend on kind of all these fertilization outcomes. So in this case, if we imagine it really to be a 3-gene system, more complicated than the original 2-gene system, you get 1 out of 64, at one end, fully white, 6 out of 64, 15 out of 64, 20, 15, 6, 1 again, right? So the curve starts to smooth out even more when you talk about the contribution of more and more and more genes. Okay? And ultimately, if you've got lots and lots of genes, you get phenotypes that can be explained that look like this rather than those discrete phenotypes. So this is the polygene hypothesis, that as you have more and more genes and more alleles contributing, you start to kind of get even finer and finer gradations in phenotype that starts to look like these normal curves. Okay, and the other point here is, so what good is this? Why are there so many genes in these wheat strains that seem to do the same thing? And it turns out it's just kind of an accident. It seems like the wheat genome, the wheat that we use today, grow today, is actually a hybrid or a duplication of multiple prior wheat genomes that occurred maybe during fertilization events and so forth a long time ago. So you have duplication of the wheat genome once or twice, resulting in a genome that has lots of duplicated genes. So here's an example where you have, let's say, 3 genes that all came from some original primordial genome that had 1 coloration, but if you duplicate it, now you get 2 coloration genes. If you have another genome added, you get 3 coloration genes and so forth. So it seems like just an accident of how the wheat genome evolved through fertilization events and so forth early on. And so you get 3 genes that just so happen to contribute a little bit to red phenotype. So that's kind of why these genes are redundant. Okay, so this slide is to highlight something that I've pointed out again and again, beginning with the personality of all those strains of pea plants like Aladdin and Banquet and all that kind of stuff. There's lots of genes. Genes have lots of possible alleles. Any given individual can only pick 2 alleles at a given gene, right? Your choices might be very large in a population in a sense, but in the end you only get 2. So if there's lots of alleles to choose from in a population, every individual is only 2 of those, for instance, A2, A4 out of a set of possible A alleles. The other point is most phenotypes are not caused by single genes or just 2 genes, as we saw in all those previous examples. Most phenotypes are the result of lots of genes acting together to produce complicated outputs like ears and toes and whatever. Okay? And if you collect all the possible alleles across all the possible genes, you get a very complicated and a very specific output. If you have a genome composed of a different set of alleles, you get a different output, right? That's the idea I've kind of harped on, that if you extend some of these ideas across the entire genome, it becomes very, very, very complicated. And if you consider lots of individuals and a particular phenotype, lots of alleles, then the curves start to become kind of continuous and can be described by these normal curves. So things like height, disease resistance, all these complicated things seem to be swayed by the particular collection of alleles that are present in one person versus another person versus another person. So for whatever characteristic, the alleles cause you to be over here on the curve, in the middle on the curve, over on the right on the curve, and so forth. And this is fundamentally the idea of quantitative genetics. Okay. Oh, we should go through some of this basic statistics of variation, even though we don't want to, okay? But we should, because some of the problems ask you to determine things like standard deviations. I gotta say, I don't really care if your calculator can just do that. I don't really want to give you columns of numbers and say, add these, can you add these together properly, and so forth. That's boring. But we have to do a little bit of this so that you can work some problems, and then we can understand the underlying genetics, which is more interesting. So, the idea here is pretty straightforward. You score a whole bunch of offspring and put them in various bins. Here we have the weight of beans. I mean, who cares? But anyway, it's the weight of beans. We have a certain number of beans in these categories. If you were to make your bins even smaller and smaller and smaller in size, you'd start to see normal curve or distribution up here, where some beans would be very light, some beans would be very heavy, and a lot of beans would be kind of intermediate. And in quantitative genetics, we ask ourselves, what's responsible for a phenotypic distribution like this? And part of the answer is the collection of alleles in the organism that is giving rise to these beans is a little different here compared to here. So you're producing a little bit smaller beans because of the alleles you have, or a little bit larger beans because of the alleles you have, or something in the middle. So part of the answer for these distributions is the allele combination. The other part will be things like environmental influences and so forth. We'll examine those a bit later. So you get some frequency distribution. This is a way of depicting phenotypic variation, and we have to have some statistical measures to analyze this. And those can be things like average or mean. That's easy. You take all the scores of exams on exam 1 and you divide by the number of students and you get some average, right? That's one thing. That's pretty straightforward. The average here is in the center. Variance is a little more complicated. It's a formula that's shown here. I don't care so much about it except to say that it's a way to describe how far out the curve extends. Is it a wide curve or a narrow curve? And there's some fancy formula like take each measurement minus the average, sum them all up, square them, divide by n-1, whatever. Okay, it's a formula. But what you get is some mathematical measure of whether the curve is really tight or whether the curve is really far apart. And the other thing that the book describes is standard deviation, and that gives you another measure of whether the curve is very narrow or the curve is very wide. The phenotypes vary a lot or they don't vary a lot. And the nice thing about standard deviation is that the units are the same. variance is squared, standard deviation, because it's the square root of the variance, has the same units as your original observation. So you may have to kind of plug and chug a few things to do problems of this type, but again, I'm not terribly interested that you know how to do a standard deviation from a column of numbers, because that's a measure of whether you can push the buttons on your calculator. I'm more interested in the kind of the genetics behind it, and we'll get to some examples of that. So here is a book example of various normal curves that are spread out differently. The mean is the same, the average is centered at 10, but these could be 3 different phenotypes that are measured in a population, like height or weight or whatever it is, and different phenotypes are either very narrow, like this purple one, with a relatively small variance, or other phenotypes spread way out and will be very broad, as indicated by this kind of orangey line with a much larger standard deviation. So these are just ways in which we can kind of analyze or measure these different phenotypic curves. So that's fine. The book spends a lot of time talking about things like correlation, covariation, regression analysis, all this stuff. We're not going to do this stuff, okay? I just want to make a brief note about correlation, because correlation is interesting and important. A lot of times you'll have one phenotype which varies, and you'll have another phenotype which varies with it. So there's seemingly some correlation between X and Y measurements. And sometimes that's meaningless, we'll see later, but other times it's important because it says that the genes, the collection of genes that are causing you to fit at some point on one curve are also causing you to fit on another curve in some related way, right? These measurements are correlated. So sometimes correlations between phenotypes tell you something about the fact that the underlying genes are having an effect both here and over here. So correlation is something that's interesting to geneticists. So will X vary with Y? There might be, or might not be, some underlying biochemical or genetic connection. Like I said, the book goes through a lot of this stuff in detail, and I'm not going to do that, so be kind of aware of that. So here's an example of a correlation, right? This is a popular thing to look at, the idea of height. Is height inherited, not inherited? To what extent it is? Is it inherited? To what extent is it dependent on environment and diet and all these types of things? One of these kind of unanswerable questions. So here we have a couple graphs showing you, in this case just a few data points, in this case lots of data points, the height of a father compared to the height of the son. Okay, so you'd like to get a sense, is there a strong genetic contribution from the father to the son that will dictate height? Okay, and it doesn't have to be height, it could be any complicated phenotypic trait, but here's the example we have. So the question is it correlated? And you can see that this line or this angle suggests that to some extent, yes. The fathers that tend to be slightly taller here have offspring that are slightly taller. The fathers that are slightly smaller tend to have offspring that are slightly smaller. And you get a sense of it here, especially with the line, and you get a sense of it here with all these additional data points. So there's some correlation, but it's not exact, right? You can pick an individual at any given spot here, and you can go up and look at the height, and you can see that there's a range at any given x-axis position here. You get a range of things here, a range of things here, a range of things here. It's just that overall you see this kind of correlation or tendency. Okay? And this has a partial genetic component, right? The father is contributing alleles that either cause greater height or don't cause greater height. And so some of those alleles are inherited by the offspring, and so you get this kind of general type of correlation here. So something we could think about, why isn't it a +1 kind of perfect correlation, right? That's a reasonable question. Why isn't every point that we see here on a line, so that for any given height here we see an exact corresponding increase in height for the offspring? And you can think about things. One is, the father isn't contributing every single allele that contributes to height to the offspring, right? The father is contributing a haploid genome, and that haploid genome is a recombination of the two genomes that he himself has. So he's contributing, some alleles that contribute to this phenotype and some not. So you don't get everything that your parent has. The other thing is there's a maternal contribution, which isn't really depicted here. So there's another set of alleles coming in as well that are going to have an influence. So that's a couple reasons why we don't see exact correlation. And there's environment, right? Environment smooths things out, causes variation as well. Things like diet, whatever. So that's going to have another effect to cause the offspring to vary a little bit relative to the parents. Okay, good. So let's see how much time I've got. Okay, let's go through some examples. The examples are more fun, okay? Some examples of quantitative genetic phenomena. So the first one has to do with corn ear length. And this helps you think about what we're trying to do in quantitative genetics. We're trying as best we can to analyze lots of genes at once to see how they produce outputs that vary continuously. So here we have a true breeding strain of corn that produce ear sizes that are fairly small. Here's a true breeding strain of corn that produces ear sizes that are fairly long. We could take these measurements and we could graph them, and that's what they've done here. You have one set of parents that produce sizes of ears that are distributed here with a mean way down here at 7 or something like this, and another true breeding strain that produces average sizes of the ears that are way up here around 16, 17. And you can see that they're spread out a little bit, and that's due to environment, okay? If these are true breeding, it means that as you breed them together to generate offspring, there's no genetic variation, right? They're homozygous at the alleles that are causing either this ear size or this ear size. So this is true breeding and homozygous for the alleles that are causing this. These are true breeding, homozygous for the alleles that are causing this. They're genetically the same. Any variation you see can't be due to genetics then. It has to be due to something else, like environment. So they're spread out a little bit, probably due to some environmental influence. Okay, this strain can only contribute a certain exact set of alleles to the gametes. Same with this one. You put them together, you get an F1 hybrid. All the F1s are genetically identical, right? If this is small a and this is big A, you only get big A, small a here. Of course, this is 10 or 100, whatever genes, right? But you only get one single thing in the F1. They're genetically identical. They produce an intermediate ear size. Why? They got half collection of alleles causing larger size here, and half collection of alleles causing smaller size here, so they're intermediate. You graph them, and they're somewhere in the center. They still spread out because of environment. They're genetically identical, but there's some environmental influence, so we see a little bit of spread due to that. And then, again, as always, we cross F1s against F1s to generate F2. And what you see is that the F2 have now spread out completely to the original point of this grandparent and this grandparent. We've gotten all the way to each end. It's more obvious in this picture than it is from the graph, but the idea is while your mean is the same, the phenotypic curve has spread way, way, way out, okay? So this is what we observe in these pictures here and in the data that are plotted here. And I want to just summarize then in the next slide or two kind of what it is we see and how we could explain this genetically. So one thing is the parents are true breeding, and I already mentioned that. We have to invoke that so that we get F1 offspring that are genetically identical. So parents are true breeding. Parent 1 has alleles for short ear. Parent 2 has a bunch of alleles homozygous for long ear. So that's fine and good. Point 2, in the F1, the alleles causing small size from one parent are combined with the alleles causing large size from the other parent. All the F1s are identical, and we get an intermediate phenotype which spreads out a little bit due to environmental influences. Okay? And point 3, The F2 average is the same as in F1, but there's much greater variance, right? You're spread way out here compared to here. And we will suppose that it's not the environment that has changed to cause something different here to here, right? We could grow everything in the same year, in the same field. We could control for environment. And yet, when we measure these F2, they still spread out much more widely. So we would like to say that when we see this much greater variance here, extending way out to the original grandparent averages, that it's not due to environment, because we do not suppose that the environment affected the F2 more than they affected these or these or these, and we could prove it by controlling for that. Therefore, the spread has to be due to assortment of various types of alleles causing short ear size or long ear size. Okay, so some notes about it. How about an explanation? Let's suppose there's a bunch of genes that come in an allelic form causing short ear size. So parent one maybe has, for genes A through H, just an arbitrary number of genes, the recessive or small letter alleles at those genes, and that causes small ear size. And let's suppose the other parent has capital letter alleles for all these genes A to H, and the capital letter alleles each contribute a little bit to slightly longer ear sizes. So this would be true breeding, short ear, this would be true breeding, long ear, we cross them, we get only one genotype, which would be a heterozygote for every one of those genes, big A, small a, so on and so forth. And so now we have kind of half the number of capitals compared to here, and half the number of small letters. So these are intermediate, but they spread out a little bit due to environmental influences. Now we suppose we cross F1 against F1, and we get a whole range of things which I haven't written out here, but here's our cross, right? And if we could treat every one of these independently, you contribute big A or small a to the gametes, big B or small b to the gametes, big C or small c to the gametes, and likewise for this one, and you'd end up with a range of genotypes that were ranging from here to here, with all the intermediate ones present as well. And that's an explanation for why we're getting this extended variance in the F2 offspring. Okay, let's see. Okay, so if this plant produces gametes and it has lots of capital letters, and this plant produces gametes that has lots of capital letters, you're over on one side of the curve. If it just so happened that the gametes produced from this plant had small letters and this one had small letters, you'd be over on the other side of this curve. What's the chance? Okay, so here's a question. What's the chance the F2 offspring would be this, which would be way, way, way over on one side of the curve as the same as the original parent? Okay, so this is the kind of question that you might consider. So the chance an offspring would be small a would be the chance the gamete from this plant was small a, which would be half a chance, and the chance that that gamete fertilized a gamete from the other parent that also was small a, so that's half a chance, right? So a lot of questions will kind of ask you what's the frequency of certain things occurring in a mating like this, and the chance of being small a would be half a chance of getting this, half a chance of getting this, right? If you extended that to B, now you would have to consider, well, there's a half a chance of getting small b and another half a chance of getting small b. And so you could do this type of calculation for almost any genotype that you would, suppose that you'd be given. Okay, so you could have questions, what's the chance a certain type of gamete would be produced from this plant? Or you could ask the question, what's the chance an offspring would have a certain genotype? In that case, you'd have to consider the gametes from here and from here. So different questions will look at this in a slightly different way. Okay, so good. Does it make sense? It's kind of a neat way of looking at things, and it expands things to multiple genes. We'll do one more example that's related to this here, transgressive segregation, and then we'll stop. Here we have a situation where the phenotypic curve for one parent is like this, with a smaller average, and the phenotypic curve for the other parent is like this with a slightly bigger average. Okay, and I've just kind of drawn these arbitrarily, but here you have two normal curves where the averages are slightly different. let's suppose that the parents are true breeding, so they can only contribute one type of allele combination to the gametes, and if you cross them, you'd always get this, cross them, you'd always get this, like in the previous example. So we cross the parents, we get an F1 that's intermediate. Nothing has changed compared to the corn ear example, right? We get some alleles from this parent, some alleles from this parent. All the F1s have exactly the same genotype. They have an intermediate phenotype due to how those alleles are adding up to produce the phenotype. We cross these, and we get a phenotypic curve that spreads way out. And in this case, the idea is supposed to show you that it spreads way out even past the original grandparents here. So the question here, which seems puzzling at the outset, is wait a minute. You have grandparents with phenotypes here and here. You generate F1 progeny that are intermediate, but then somehow you cross the F1s and you get phenotypes that are way outside anything you've seen in the past 2 generations. So the question is, how could we explain something like that? Transgressive segregation, where you seem to be going way out here on this phenotypic curve in this direction and this direction. And the answer is pretty straightforward. Let's suppose that one of the parents is true breeding, small a, big B, small c. So 2 capital letter alleles contributing to some phenotype. And let's suppose the other parent is big A, small b, big C. 4 capital letter alleles contributing to some phenotype. We could assign some value for each of these capital letter alleles, like 5 for whatever measurement it is, right? So in this case, the phenotype would be 2 times 5. In this case, since there's 4, the phenotype would be 4 times 5. So parent 1 would be 10, parent 2 would be 20 for whatever the measurement is. That's why parent 1 would be here at 10, let's say, and this would be here at 20. Right? So it's kind of theoretical, but you get the idea of how we're thinking about the incremental additive effects of genes in quantitative genetics. So now, we breed these together. What do we get? We get a heterozygote, which is going to be intermediate in terms of the number of capital letter alleles. We get a big A from him or her, we get a big C from him or her, and we get a big B here. So the offspring F1 are going to be big A, small a, big B, small b, big C, small c. 3 capital letter alleles, assume we give 5 points each, we get a value of 15. So the F1s are intermediate, so fine. Right? Now we cross the F1s, but when we cross the F1s that are heterozygous at these 3 genes, we can go all the way from only small ABC, all the way up to all capital ABC, right? And if you start multiplying, 0 capitals times 5, you get 0, so no contribution. Or you multiply 6 capitals times 5, you get 30. And that's why in these F2, we can see this transgressive effect where the grandkids are having phenotypes that have spread out even further than what they saw in the past 2 generations. So kind of a neat little thing. The idea here is the same as the corn ear thing, and both of these examples highlight the idea that we're thinking about tens or hundreds or thousands of genes at once, which are adding up a little bit here and here and here and there, depending on the collection of alleles, to cause a phenotypic curve where you'll be placed on one side or the other side or in the middle or something like that. Okay, so that's good. Have a good weekend. We will continue discussion of